right, welcome everyone. Thank you for joining us today on this discussion about the role and future of cancer vaccines in immunotherapy. We have a very distinguished set of panelists and a diverse set of platforms, technologies. So I will let each of the panelists introduce themselves and talk a little about their company, and then we'll get into the roundtable discussion. And again, I'm Jeff Bockman, EVP, and I lead the oncology practice at Cello Health BioConsulting, previously defined health. So I will go, as I see on my screen, upper left, Carlos.
Hi, everybody. It's a pleasure to be here. My name is Carlos de Sousa. I'm the CEO of Ultimovacs, a biotech company based in Oslo, Norway. We are a cancer vaccine company. Our lead product is a next generation telomerase vaccine. We basically have a universal vaccine that educates the immune system to target cells expressing telomerase, and 80 to 90% of cancer cells in solid tumors express telomerase. We are currently in phase 2 development. We have 4 phase 2 trials with more than 500 patients in melanoma in combination with ipi and nivo, in mesothelioma, also in combination with ipilimumab, nivolumab, this one in collaboration with Bristol Myers Squibb, in ovarian cancer, in combination with olaparib and durvalumab. The study is a collaboration also with AstraZeneca, and the 4th study in head and neck cancer, where we combine with pembrolizumab. We will also be communicating data now at ASCO from our phase 1 to 2A study in malignant melanoma in combination with pembrolizumab.
Great, thank you, Carlos. We'll go to you next, Fred.
Hi, everyone. My name is Frederic Ors. I'm the CEO at IMV. We are developing T-cell therapy based on a drug delivery platform that basically allows us to greatly extend the T-cells we can generate in humans. With the first asset, we are in multiple phase 2 clinical studies in 6 different indications, and we've already generated positive clinical results, both as monotherapy in ovarian cancer and as combination in lymphoma in relapsed refractory DLBCL, and that in that case, it's a collaboration with Merck, combination with pembrolizumab. We are, based on that, we are moving into late stage phase 2B trial, the first one being in lymphoma in relapsed refractory DLBCL. We are initiating the study in this quarter, and are developing other assets at earlier stage of development in breast cancer and bladder, for example.
Great, thank you, Fred. I'm going counterclockwise. So Andrew, you're next on my screen.
Great. Good morning, everybody. Thanks for inviting me to join this panel. Pleased to be here with this distinguished group of colleagues. My name is Andrew Allen. I'm the CEO, one of the founders of Gritstone Oncology. We're a publicly traded company, about 6 years old now. We believe that prediction of T-cell epitopes is important and hard. So we've built a key machine learning platform for taking sequence data and identifying with high accuracy which mutations will actually form neoantigens or antigens for your immune system, particularly the CD8 class 1 presented antigens. And then secondly, we've built a vaccine vector system that then can elicit in higher species, including humans, a very strong CD8 T-cell response. And we've applied this pair of capabilities to cancer initially, so neoantigen-based cancer immunotherapy, and we're now in phase 2 studies. We're completing a phase 2 study in very advanced disease patients, a classic kind of phase 1-2 study in, let's all agree, I think the toughest patients to treat with cancer vaccine-based immunotherapy. And encouraged by the signals we've seen, we're now launching randomized phase 2 studies in earlier disease settings. And we'll be sharing data at ESMO, both in terms of the output of the phase 1-2 study, as well as the plans for the randomized phase 2s. We've also applied these technologies to infectious disease, and we have a collaboration with Gilead on an HIV vaccine, and also have a COVID-19 vaccine program, which is currently in phase 1 testing by NIAID, the Dr. Fauci's team at NIH.
Great. Thank you, Andrew. Nicolas.
Hi, I'm Nicolas Poirier. I'm the Chief Scientific Officer at OSC Immunotherapeutics. So we are a clinical stage French biotech. We developed immunotherapies in immuno-oncology, autoimmune disease, and transplantation. So we use monoclonal antibody platform as well as our cancer vaccine or CD8 T-cell neoepitope vaccine platform. Our lead project is therapy, which is currently in several phase 2 combination studies with Nivo and Pembro. And we announced nearly 1 year ago our positive step 1 result of a phase 3 trial in monotherapy in non-small cell lung cancer after failure to immune checkpoint, where we showed increased efficacy in terms of survival and benefit for the patients in terms of less reduced adverse events. And we also, similar to Andrew, launched a phase 1 trial of COVID-19 prophylactic vaccine using this platform, targeting 11 targets of the SARS-2 virus to cover all the variants.
Great, thank you, Nicolas. And lastly, Mike.
Hi everybody, great to be here, and thank you very much to the panel for the invitation and the chance to discuss with you all today. My name is Mike Mulqueen. I'm the head of business development for eTheRNA. We're a small Belgium-based biotech company working in the mRNA field. Similar to many of the other panel members, we're developing vaccines both for cancer and for infectious diseases, including, of course, COVID, because everybody in RNA has to have a COVID vaccine somewhere in their portfolio. One of our novel technologies is an mRNA-based adjuvant, which is capable of activating dendritic cells and stimulating them, maturing them, to enhance their presentation of antigens to CD8 and CD4 positive T cells. We've achieved proof of concept for this adjuvant in multiple melanoma using an ex vivo clinical study design, including 2 phase 1 and 2 phase 2 studies, which demonstrated significant patient survival. So we have confidence that this adjuvant approach in combination with tumor-specific antigens can lead to a very promising therapeutic benefit. We similarly have used neoantigens in some of our other cancer vaccines and advancing those towards the clinic this year. We believe that the mRNA field in particular, but all the RNA technologies we're going to hear about today, are at that interesting cusp of just breaking through into real therapeutic benefit. And I think, terrible as it's been, the COVID pandemic has really demonstrated the strength of the platforms. And we're hoping to build on those kind of results in the future for both cancer and infectious diseases.
That's great. Well, thanks, everyone. So I'm going to start with the, as we all call it, the elephant in the room about cancer vaccines. And that is, you know, they are one of the earliest, one of the longest standing, and one of the most baggage-laden platform technologies in cancer, let alone in immunotherapy. So I'd like to hear thoughts from all of you about, you know, not only why that is, but when you all feel, or when each of you feels, we'll actually kind of get true validation. And that's not only speaking to, you know, your belief in the strength of your own platforms, but broadly looking over the field. So anyone want to jump on that first? Or I can pick on someone.
I don't mind having a go.
Sure, Mike, go ahead.
Okay, so I think, yes, it's definitely the big question mark. When will we be able to demonstrate that the cancer vaccine theory really falls out? I think Andrew mentioned right at the beginning that we tend to have a habit, I think, as drug developers, that when we get a really new promising technology, we immediately point it towards the most difficult to treat patients we can possibly ever get and really put up a high hurdle for its efficacy. And I think maybe that's happened to some extent with the cancer vaccine. The ideas have been great, but where they've really suffered is trying to identify those specific antigens that will allow the unique targeting of the tumor, cover a wide variety of different tumor types, and within even one tumor, there are several different cell types, and still differentiate away from normal tissue. So you have the high safety and the high efficacy burdens. I think more recently, the advent of technologies like RNA has enabled us to express antigens that previously were not really accessible to the immune system, and to express them in a way that can activate that immune system. And in conjunction with other combination therapies, such as the checkpoint inhibitors, I think we're beginning now to see the validation of the whole cancer vaccine approach. As I mentioned during my introduction, we have demonstrated using an ex vivo approach, a very potent ability to protect multiple melanoma patients against cancer reoccurrence. So I do think that we are beginning to see the maturation of both the technology and, perhaps as importantly, the clinical designs that enable us to actually validate these ideas.
Thanks,
I agree with all of that, Michael. I think you say it well. I think it's always good to just recognize and be humble that when we started cancer vaccinology, we just knew very little. And we did the best we could with the knowledge we had, but it obviously wasn't good enough. I think we've learned a lot, both through cell therapy, through checkpoint inhibitor therapy, and today we can articulate a therapeutic hypothesis that is much more crisp and better informed than we used to be able to do. So I would say that today we know that neoantigen-specific CD8 T cells are capable of driving tumor cell destruction and long-term clinical benefit. I think the checkpoint data, next-gen sequencing as a technology, a lot of sophisticated immunology has really provided the scientific support for what I just said. So if you take that belief and then say, well, where do we go from here? You know, I'm pretty confident that most of us could probably vaccinate patients with melanoma and drive some form of additional clinical benefit, but it might be pretty modest because those patients typically already have a neoantigen reactive CD8 population. We may be able to add to it, add a few more antigens, or we may grow a few more T cells, but fundamentally we're moving the needle very little in that context. So I think it's going to be more straightforward for us to work in areas where current immunotherapies don't do very much. But we believe the bones are there, i.e. let's find tumors that have neoantigens where the deficit is that there is no existing adaptive immune response to those neoantigens. There are no T cells. And if I treat those patients with a checkpoint inhibitor, all I will do is probably induce autoimmunity because the substrate isn't there. That's the group where obviously with the vaccine, you can elicit neoantigen reactive CD8s and potentially really change the trajectory of their disease. So that's the way I think we increasingly are thinking about using our technologies and applying them to patients in a way that's potentially more meaningful. And to finish on Mike's point, treating end-of-life subjects who've got beaten down immune systems, highly complex tumors with lots of genetic heterogeneity is just a tall order. And often it takes 8 to 12 weeks for us to see strong CD8 responses. And some patients are progressing and dying within 8 weeks. Well, clearly those are patients we're never going to be able to help. But that's the way the phase 1 system operates, fair enough. You have to sort of survive through phase 1, hopefully see signals that mean something, then move into earlier stage settings where you have time to do all of the good things that our products can potentially do. So I think what we've learned is important. It's key that we learn from it and that we reformulate our hypotheses and test them using current knowledge.
Yeah, and so between the two of you, Mike and Andrew, you've kind of framed a lot of the classical issues raised about, the kind of the history of cancer vaccines for those who still truly believe in their power and have to counter those who are naysayers in terms of, which type of patients, earlier stage, the nature of, as we might describe it now, per Chen and Melman, kind of the idea of the inflamed versus the excluded versus the desert type of tumor types, the use of and relative to checkpoint inhibitors. So a lot of the key points are not only germane to the underlying kind of biology, but also to the clinical development strategy. Anything you want to add to that, either Fred or Nicolas?
Yes.
Go ahead, Fred.
Go ahead, Fred.
Okay. Well, just I want to just add one thing that I believe is true for cancer vaccine, but I would argue probably for a lot of immunotherapies in development in cancer. Even if we've learned a lot of things from checkpoints, I think it's still, kind of a black box in the middle. You have the input and the output, and what's happening in the middle is very difficult to figure out because of the complexity of the immune system. And so developing a conventional drug development pathway in clinical is very hard because of this. And what we're trying to do really, and we hope we can contribute to the validation of the concept of cancer vaccines, is to bridge a little bit of that gap by really focusing on working on the pharmacokinetic and pharmacodynamic of the T cells, like in a more conventional way, in a way that people are used to, where we can relate the biological activity, so tumor regressions and clinical responses to the dose of T cells we are generating. And by proving the biological activity, maybe participate into the validation of the concept, whether it's in late stage or early stage, relying on that biological activity. And one thing we do that help us reinforce that idea is that we are targeting targets in cancer that have a role in the biology of cancer, a bit like targeted small molecule drugs are doing, so that you can again relate what you are seeing happening in the tumor, the regression, to the evolution of the biology of the cancer that you are treating at the same time. And you can do a more complete, we believe, demonstration of the power of this technology is having now, but also can have in the future.
Great. There are a couple of points I want to pick up in what you said, but I want to give Nicolas a chance to weigh in.
Yes, I think we are still hunted by the failure and of previous big trials in the field of cancer vaccine before the revolution of immune checkpoint. But what we think also is that maybe we have to change habits and on how we can recognize a benefit for our patients. For example, if you only look at tumor shrinkage, this is maybe not appropriate for the cancer vaccine. We have a delayed but a long-term effect. And for example, with chemo, you can have an initial very quick tumor shrinkage benefit for the patient, but then the patient quickly progress. While what we have seen with the cancer vaccine is a long-term benefit, long-term protection, and clear correlation between is the level of immunogenicity that you can capture, monitor in periphery, and the duration or the increase in terms of survival for stage 3 and 4 patients. So maybe we have to change also that point in terms of clinical developments.
And thank you for Nicolas, because that exactly was one of the points I wanted to pick up on that Fred brought up and that we talked about in the prep call is, this issue, whether it's a challenge, whether it is a disconnect, whether it's inherent to the nature of vaccines, I'd like everyone to talk about, and that is this aspect of kind of response rates, whether resist or iResist or otherwise, versus outcomes, whether PFS or time to tumor progression or OS, et cetera. So who wants to kind of expand on that next? Maybe you, Andrew, because I know we've talked many times about it historically.
Yeah, for sure. Nicolas raises a great point. We all understand that RECIST response rate for solid tumors has been historically a pretty good metric for whether a therapy works or not. And I think people have become a little bit lazy, and that's all they look for. But I think it's good to remind ourselves of two things. Number one, it is not clinical benefit. And you have this strange situation where if we had two patients, one of whom was offered a therapeutic that gave them, quote, stable disease per RECIST, and they were alive and well at 12 months. And another patient could have a response at cycle 2, the end of month 2, that lasted for 6 months. Then their disease recurred, they progressed, and they died at 12 months. I think we all know which one a patient would want, but you would never get to that first therapy because you would be regarded as ineffective because I'm not seeing response at cycle 2. So I think we need to step back and remind ourselves that we're in this to help patients, and patients care about living a long time. And sometimes RECIST response at month 2 is a useful index of that, and sometimes it just isn't. And what we're trying to accomplish with these vaccine-based immunotherapies is fundamentally likely to not lead to RECIST response at cycle 2. Because of course, RECIST assumes that you have a single cell population in tumor lesions, i.e. tumor cells, and your therapy will kill them and they will disappear. And the lesion will therefore shrink. And that works when you've got cytotoxic chemo or a targeted therapy. What we're trying to do is fundamentally different. We're trying to drive a new cell type, T cells, into the tumors where we hope they will meet their antigen and proliferate. So we're trying to cause expansion of lesions in some sense. Now, of course, when you look at a CT scan, you have no idea what the relative mix of tumor cell and T cell is. But we know from the checkpoint data, certainly in melanoma, that pseudoprogression, i.e. lesion expansion at month 2, is pretty common and leads to good outcomes. And that's when the T cells already exist. So, checkpoints generally work when there already are the CD8s in the tumor and you're causing their expansion. What we're doing with vaccines is obviously way proximal to that. We have to generate the T cells, which then have to traffic to the tumor cell, to tumor lesions, and hopefully then expand. And so, frankly, if you saw lesion disappearance at cycle 2, you'd be perhaps a little bit surprised. So the focus on early RECIST response, I think, is completely misplaced for vaccine immunotherapy. Now, over the long term, you may see lesion disappearance. That's possible. We will find out as we travel. But this myopic reliance on RECIST at cycle 2, I think, is fundamentally a mistake when you're trying to discern the effectiveness of vaccine-based immunotherapy.
Well, and let's broaden that, because that's related to, and I'm going to ask this of you, Carlos, and then you, Fred, because this relates also to something that Fred brought up, and that is the nature of the type of antigen, and maybe the distinction about going after kind of driver mutations, which may be more like kind of targeting, the tumor more specifically, in which you perhaps might expect some type of ORR. So I'd like to hear your thoughts, Carlos, in terms of kind of where you're coming from, at least with the UV1 telomerase vaccine, and I'd like to hear your thoughts on that as well, Fred, in terms of this RECIST and ORR aspect versus kind of the outcomes in terms of PFS or OS. So, Carlos?
Yeah, I will start there because it's a very good point. In our first phase 1 studies, we had patients that progressed, and the investigators just decided to continue the treatment. And these patients, by RECIST, would have been classified as progressors and basically leaving the study. And a couple of them then moved to complete responses. So in this is, I think, the drama a little bit is how many patients are we missing in terms of efficacy and kind of leave the studies and lose the benefit just because of the measures. So we definitely see, and also in past lives, it's clear that vaccines and other cell therapies, the benefit comes late, but then is a long tail. So how are we going to capture that? That's maybe we need to change a little bit of the measures in clinical trials of really RECIST not being the drivers to decide if a patient is progressing or not. Honestly, I don't know what we can do, but I think that's a big challenge, and we are missing potentially some of the efficacy just because the time when we make the assessment. Regarding our approach, I think Andrew mentioned, and all of you mentioned in terms of the biology. I think one cell that everybody has been forgetting about, is the CD4 positive cells. That it was CD8, and now more and more is recognized that it's not just CD8. You also need CD4, and I think this is just a recognition that our knowledge of the immune system and how it works, it's still evolving on a daily basis. But I think we are getting smarter, and I think we are going to have vaccines that have an impact. We have a benefit versus other approaches, because as telomerase is primarily expressed by the cancer cells, and this is their immortality mechanism, so they will always be expressing telomerase from early stages to late stages, regardless if it's the metastasis or the primary tumor, regardless of what part of the primary tumor area it is. So we have it a little bit easier in that regards, because if the cancer cell stops expressing telomerase, basically they will naturally die. So for us is a very good target, and we have the data to show it. But we know that there will be other areas where we can enter in combinations, not just everybody's combining with checkpoint inhibitors. I think there are other approaches that will start coming where, different mechanisms, even in terms of the vaccines, the point in time where you interact with the immune system and what you stimulate are going to open new avenues in terms of combinations that at the moment, by the nature of our business and the challenge for small biotechs is, of course, everybody starts with checkpoint inhibitors, but I think we're going to start seeing more and more of triple combinations, or even more, just because the immune system needs all the different types of approaches to really, have the efficacy that you need.
Yeah, I'm going to come back to that later, but Fred, I wanted to give you a chance to weigh in too, in terms of your point about the driver mutations and.
Yeah. Thank you, Jeff. First of all, to me, I think I totally agree that the unique value proposition of T-cell therapy cancer vaccines is that long tail of duration of clinical benefits you can offer, and in the absence of significant toxicity. And that's what we have to offer at the end, and hopefully the validation will get us there. I don't necessarily see that I believe you can have that. You can also have biological activity, and we've made demonstration of that. They are not mutually exclusive. If you are in a patient population that are progressing fast and you need to stop progression, you need to impact the tumor early to be able to keep the patients on treatment, because it's very hard to make differences between patients, benefiting beyond progression and the others. And I think T-cell therapy and cancer vaccines can do both. I think it's possible. It depends on the indication you're going on, and also I believe if you can go after a target that is involved in the biology of this specific patient subpopulation, then you have a better chance of impacting the evolution of that cancer. And on top of that, you get I don't know if we should call that prevention, but it's some sort of prevention you're getting from the immune system that prevent the recurrence or prevent the patient to progress too fast. And that's where you get the meaningful benefit and the improvement of the quality of life.
Yeah, I'm glad you mentioned that. We're at least internally, colloquially refer to that as kind of secondary prevention, in line with, cardiovascular, et cetera. But Mike, I'd like to give you a chance to kind of chime in on this part of the discussion.
Yeah, I think the whole field is advancing as we learn more about which antigens really are the right ones to target. I think one of the early ideas behind the cancer vaccines of kind of converting those cold tumors into hot tumors was let's take something that flags a tumor cell. It doesn't really need to have a function itself. It just needs to be able to flag that this is a tumor cell that the immune system needs to attack. And once you've started that immune system going, other antigens will emerge and the immune system will escalate, and eventually you'll get a clearing response. But I think actually choosing the antigens much more, using bioinformatics and other techniques to actually identify the most relevant antigens, rather than just the one that happens to flag a couple of tumor cells, is probably actually going to be the much more sophisticated approach. I remember when I first started out in immunology, which was a long time ago, the immune system was simple. You had the innate immune system with polymorphs that dealt with short-term infections, and then you had the sophisticated learning immune system, which was B cells and T cells, and that dealt with everything else. And then, of course, it gets much more complex once you start looking at helper cells and CD4s and CD8s and delayed responses. But in reality, the immune system is one system. And we artificially divide it up into these different cell types and different phases of response. That's not how it happens. And I think there's a big push now that the cancer vaccines in promoting T-cell responses are only part of the answer. You might actually want to promote polymorph responses. You might want to bring neutrophils in at some stage. You might want to activate all different types of systems to deal with these cancers. And I think we're only beginning to scratch the surface of what we can do there.
Well, I'd like to pick up on that and also go back to the discussions about the CD4s and CD8s, because certainly, even when I was in graduate school with Marianne Koshland teaching immunology at Berkeley, the importance of CD4s was well recognized. I think the issue is that, historically and even in recent history, a number of the vaccine approaches, were emphasizing their correlative data, partly to counter detail against the lack of actual clinical benefit. And a lot of that often turned out to be seeing CD4 cells, and so they get to be the scapegoat, right? Whereas, it must be all CD8, and so we swing from one extreme to another. But I think your point about the whole diversity of cell types that are part of this immune system, and the fact that it's also not a clear and clean demarcation between innate and adaptive, right? There are things that straddle and cross over and even change their behavior in context. So maybe we can continue that discussion a bit, Andrew, because I think it is important, because we still, when talking with, certainly large pharma, you still see a lot of resistance about cancer vaccines. And a lot of that, is either residing in, old knowledge, like, you're really only mostly getting CD4 cells, or you may be getting immune correlate data, but no one's shown the, definitive kind of clinical data that really needs to be relevant. How do you address that at this point? Obviously, you all each with your platforms are hopeful, and I hope for all of you, because it'd be better that all of you kind of make success in ultimately getting those responses. But in this interim period where we're very close, and hopefully it's not like Zeno's paradox, where you never cross the finish line, how are you addressing those questions or concerns that come up from either pharma or investors or others? So Andrew, I'll toss that to you first.
Yeah, so data is all that matters ultimately. And so we have to generate the right data. And of course, we need to then be in the right context in order to be able to generate the right data. And we discussed that earlier, that moving upstream is where we're all heading. And then we're going to be looking for evidence of clinical benefit, because that's really what everyone cares about. The safety side, of course, is very important, but I think we can all agree that there are unlikely to be major safety issues with neoantigen-directed vaccines. So let's focus on efficacy as the reason for failure historically. And so we have to show good data. And again, we spoke earlier about defocusing from early RECIST. And some of our pharma brethren, they like it too, because it's easy. I mean, we all like it when it happens, but if it's not there, that doesn't mean your product isn't working. So how do you show them that it's working? Well, I think you have to do designs that make sense, and you have to use endpoints that make sense. So the endpoint that we've become very focused on is circulating tumor DNA. It's obviously becoming increasingly well validated as a very direct metric of tumor destruction. And we showed some data in July of last year suggesting that what we were seeing with some of the cancer vaccines was a little different from checkpoint inhibitors. So I mentioned earlier pseudoprogression with checkpoint inhibitors. And again, the biological context is distinct. So I think the idea for a checkpoint is I've got a melanoma patient who has a neoantigen reactive CD8 population infiltrating into the tumor at baseline. Those CD8 T cells could be good, but they've been shut down through PD-1 pathway stimulation. When I block PD-1, those T cells go straight back to work. And what I observe is sometimes this transient proliferation manifests by lesion expansion and so-called pseudoprogression. But what I see in ctDNA is that the ctDNA starts to go down. And the meme that's out there is that ctDNA can differentiate between true disease progression, where the ctDNA will go up, as well at the same time as the lesions are expanding, versus pseudoprogression, where the lesions will expand, but the ctDNA is going down. So that's the basic idea. Looks pretty nice and makes sense. What we've observed is different. What we've observed is that at week 8, we see lesions getting a little bit bigger, but we see ctDNA going up. We see a spike in ctDNA and actually a spike in tumor markers as well, like CEA. But then after that week 8 spike, then everything starts to go down. And I think that speaks to the different kinetic, that we don't have T cells at baseline. We have to generate them first. We get a wave of CD8s, and those CD8s go to work causing tumor lysis, this apparent sort of biochemical pseudoprogression linked in with radiological lesion expansion, and then everything starts to head south and goes in the right direction. So it's fundamentally different. So ctDNA for us looks to be pretty important. And of course, the data are now accruing, showing that if ctDNA basically goes down, that is a uniformly good thing, right? ctDNA staying flat is probably good compared to the worst outcome, which is ctDNA goes up. There's just lots of data now in solid tumors suggesting that those simple gradations of truth. So ctDNA, I think, is super important. You need to do randomized studies because obviously when you're looking at time to event endpoints like PFS or OS, single arm studies are essentially uninterpretable. Now that's not uniformly true, but you have to be very cautious. And so I think randomized studies are the simpler way to go because our field, I mean, the whole cancer therapy field is littered with therapeutics that showed a single cycle expansion in median PFS. In other words, I go from a median PFS of 2 months to a median PFS of 4 months in a single arm study. So it looks good, but it's really just one cycle difference. And as soon as I subject that to the heat of a randomized study, that apparent difference evaporates. And that's true of all cancer therapeutics. So we've got to be particularly stringent with cancer immunotherapy because frankly, the suspicion is higher, right? The onus is on us to really show definitively that this thing is working. And so randomized studies using the right endpoints in the right patients, I think are the way forward.
Anyone else want to amplify or?
I could, yeah. So I fully agree with Andrew. The data first, clinical data clearly are first, and also how we can link, biological modification with clinical benefits. And I agree with in our randomized and blind phase 2 and phase 3 studies, where we compare immunotherapy, the cancer vaccine versus the chemo. Coming back to the previous point, if we only look at the resist or tumor shrinkage, clearly chemo win. But what we observe is a very high proportion of very long stable disease, 3, 4, 5 years of stable disease, no progression. And including in patients who progress, also an increase in terms of survival compared to chemo. And what was interesting, and I think what is key also is to show that some patients have not good immunogenicity after vaccine, 15, 20% in our case. And these patients have few or no benefit in terms of survival, while the others that have high immunogenicity have clear benefit with very long stable disease. So these type of data are key. And coming back also to the point on hot or cold tumors, of course, we have access to biopsy of some lung tumors, and we anticipated that we can see cold turning out, tumors after vaccination. We were surprised that several responders, or at least patients with long stable disease, have initially hot tumors, but fully exhausted. T cells express PD-1, TIM3, LAG3. And after vaccinations, the tumor is still hot, but the T cells did not express PD-1, LAG3, and TIM3. So it seems that the vaccination could work also in the field of hot tumors and create a renew on fresh T cells that explain probably the clinical benefits. So I think this type of link are very interesting also to convince the pharma that the field is moving and interesting for the future.
Go ahead, Cross.
I was just going to say, I think part of the challenge that, when we meet with pharma, and again, we probably are within our fields more on top of the science developments is the requests, so, can you show me monotherapy efficacy, so what of course, means that, they really, in a lot of cases, they don't understand, the mechanism and the synergy that we need to have in terms of vaccines, that they are just primers. You need the other, the checkpoint inhibitors, for instance, so the other ones is, Andrew touched it, is, how can we measure that we're having efficacy? We get the question, how can you detect, these cells in circulation Yes, we can, but then sometimes the cells disappear, and but then that is bad. And I say, no, it's not bad. It's because these cells are now in the tumor. But the interpretation is that there's no cells in circulation, so so then there's no effect. So this is this kind of of a challenge that we have in terms of explaining how these things are measured, and of course I'm not even bringing the fact that do you have preclinical data? What of course is always a challenge for any of us, in terms of preclinical data when we are dealing with human immunology and cells. How are we going to get these models in animals that are true, useful in terms of our activity? So I think that. I think we also need a little bit more education in certain groups of some of the big pharmas that are not so close to this field, in terms of how all this works, so they don't come with unreasonable requests.
Yeah. Well, and of course, while I hopefully we would take people at face value, perhaps some of those are kind of false obstacles, because there's a lot of things out there to choose from, like lots of different modalities and ways to go, and some people have already fully committed, say, to adoptive cell therapy of various sorts. Others have not. Some have committed fully to bispecifics of various sorts. Others have not. Some have waded into oncolytic viruses. Others have not. So I assume cancer vaccines will continue to kind of parallel that. I want to pick up on something that Nicolas alluded to, and it also amplifies on the ctDNA that Andrew mentioned, and that is, the ability to monitor patients and to either predetermine who might be more responsive or to track over the course of time who is responding. And that might have implications in terms of, you mentioned, I think it was you, Carlos, about treating kind of beyond progression and then having people convert to CRs. So maybe we can talk a little about that. I mean, obviously there's the more, I call it basic in quote, kind of hot, lukewarm and cold tumor issues, but there are a lot of other aspects that could be mined, whether those are specific analytes or signatures, et cetera. So I'm just curious, beyond ctDNA, kind of where you think the field is going, both in terms of kind of monitoring and even perhaps pre-stratifying who might be more likely to respond. Anyone want to take up on that? And some of this may be based on the antigens too. I mean, obviously if it's a neoantigen and it's patient specific, that's one thing. If they're shared, that's also a different thing. That gives you a certain stratification, but in other cases that may not be inherent in the target.
Yeah, there's a biological circularity here. I think that, chasing hot tumors, again, you've got T cells in those tumors. They already have an adaptive immune system that has recognized tumor antigens and has mounted an immune response. And then it gets shut down by presumably various checkpoints. There's obviously a ton of interest there, and people are kind of pouring in with their TIGIT antibody, their LAG-3 antibody, et cetera, et cetera. And maybe we can squeeze a bit of additional benefit out. But if our thesis is correct, I think we should be able to help patients with cold tumors who today really get no benefit from the current crop of simple checkpoint inhibitor immunotherapies. Again, the idea would be that there are neoantigens, but the tumor has successfully hidden from the adaptive immune system, and therefore they won't respond to checkpoints. There is no substrate. So it helps if that's true, because you can then show clear signal. Because if, in colorectal, which is a tumor type we've spent a fair bit of time on, standard microsatellite stable colorectal cancer does not respond to checkpoint inhibitor therapy. Huge trials have been run, phase 3 trials with monotherapy, with chemo combination therapy, with doublet PD-1 plus CTLA-4 antibody therapy, and nothing is proving interesting or successful in this context. And so those patients just do poorly. They get 2 different forms of approved chemotherapy. Some will then maybe get regorafenib in the 3rd line, but there are no good actors. It's sort of depressing to see the data coming out of colorectal. And in principle, we should be able to help there because those patients probably have neoantigens. And obviously that's an area that we focused on. And if we see something, it should be very easy to discern that signal because the backdrop is so uniformly unimpressive and disappointing. So I think that's the context where we should be able to see a benefit. And of course, those patients really need help. So it's a good area to work in if we believe in the thesis. Now, there may be something else we don't understand. There may be some inherent reason why colorectal cancer doesn't respond to immunotherapy. But the MSI data suggests that's not true. Right? Those MSI tumors, they're in the colon. It's got the same microenvironment as far as we know. They just have a lot more antigens. And a lot more immune activation. So that's encouraging. So I think pursuing cold tumors with vaccine-based immunotherapy is potentially an important path forward for us.
Yeah, I'd like to link. Does anyone want to amplify on that? Because I want to.
I just want to say, Jeff, to me, along with the team of how can we better design clinical trials to improve the chance of success, I think if there's any way you can target immunotherapy into a subpopulation of patients that have a higher chance of responding, to me, we believe that it's really important to try to do that so that you increase the chances. And we are spending a lot of time and resources into studying, all the potential factors that could influence the response to immunotherapy when it's used as a standalone or even in a combination with a checkpoint. I think that's the way cancer vaccines could improve what was done in the past, and certainly an area where we are doing a lot of work.
So I'd like to tie some of those themes together, the cold tumor or even lukewarm, which we like. The question you posed right at the beginning, Carlos, or proposition of other combinations beyond checkpoints, and then what you were just saying, Fred, is like when you think about going into let's just call them colder, whether they're truly ice cold or sort of cold or lukewarm. How are you thinking about what combinations might be beneficial? You still might unleash, you still might need those checkpoints, which now might be viable and applicable in the cold tumor if you've got antigens and CD8s there to recognize those antigens. But, ultimately you want that immune response to spread to other antigens, other epitopes, et cetera, and recruit other cell types. So how are you thinking about what combinations should be viable with your approaches above and beyond a checkpoint? Anyone want to leap in?
I can start with one idea. Of course, the immune checkpoint are very interesting to go in a sequential approach after or in parallel to vaccination. But beyond this one, I think personally that we are good with all our platform to educate T cells, to recognize antigens, to amplify this pool in periphery. But then we have to make sure that the T cells are able to migrate in the tumors. So we have to help, but probably the T cells in terms of arming them, in terms of integrin expression and so on, to be able to migrate into tissue and to stay in this tissue, and also to be recruited to the tumor site. So probably there is room for chemokine approach locally in the tumors to recruit the T cells. We have also evidence that some cytokine are very good to prime the T cells for migration in mucosal interface, such as interleukin-7, for example. Very good cytokine to prime these T cells, able to express the integrins and migrate, for example, in the lung. Otherwise, if you prime and the T cells stay in periphery, you have good ELISpot response, but you have no clinical benefits. So I think there is room for this type of approach to in the future.
Great, thanks. Anyone else? And I think.
Oh, sorry, Jeff. I just wanted to say, share our own experience with this. We've done different combinations with Insight and Merck, and what we've learned along the way, and I think probably the true at the end is that it depends really on the indications. There are, the subtype. There are some indications where, for example, a combination with a checkpoint makes a lot of sense, and others where you're probably better looking at something different depending on that subtype, for example, and that's how we are looking at it on a per indication basis, really, to figure out what could be the best combination.
Yeah. And we haven't talked much. I mean, we've alluded to the antigens. We've touched on the platforms, heterologous prime boost, although you didn't actually directly say that this time, Andrew, but obviously RNAs that are being used, liposome encapsulation of kind of specific antigens like driver mutations or survivins and telomerase, et cetera. But what about adjuvants and their role? Because we often get asked a lot about that by clients who are exploring new platforms and thinking about ways to kind of go kind of beyond the standard. And I know a number of you have some interesting novel types of approaches to the adjuvant, which is a very vague term. So feel free to define it as you see appropriate. I see you, Carlos.
Yeah, I may start because I didn't mention, but we have a second platform. We just moved now into clinic, where we have a total novel way of basically doing that. We have a core that works as an adjuvant. We call the TAT platform. Is uses the antibodies already present after the tetanus vaccination, and basically in the same molecule we have the adjuvant, and we have the peptides, or anything is really is almost like a kind of a cartridge where you have the nucleus, and then we just mix depending on the type of tumors, and I think this is a different approach and takes us away from some of the challenges of using the GM-CSFs or other type of adjuvants that can create challenges, and we saw that also now with even some of the challenge with the COVID vaccines, where the adjuvants were in some cases the challenge. So I think we definitely we are working there, and we are very excited to start getting the clinical data, but I think we definitely need to look for alternatives and to have this adjuvant activity out of the traditional ones.
Great.
Yeah, and I would certainly agree with that. We have an adjuvant system which is based on mRNAs for 3 specific molecules, which are co-expressed alongside the tumor antigens. So we have a local expression of the adjuvant alongside the expression of the antigen of provoking the vaccine response. And I think that gets away from some of the problems of a systemic adjuvant, as it were. We are learning that depending on the nature of that adjuvant, you can actually train the immune system down particular pathways. Certainly, you can, for instance, increase expression via the MHC2 pathway rather than the MHC1, which can provoke a different type of immune response entirely. So I think increasing knowledge of the adjuvant side of it is going to help to educate the immune system to the systems that we need it to be. One of the things that does make me concerned, though, is that we can't, Andrew mentioned this early on, we're treating often patients who are in end-stage disease and don't have the time for us to experiment with what the best combination of things is for them. So we need better systems for predicting response rates that doesn't rely on whether the patient is alive 6 or 12 or 18 weeks after we've dosed them. And I think that's where we're still learning how these different systems interact with each other. And all these ideas about combining cytokines and chemokines, which I certainly agree with also. Often we don't have the clinical time to do that. We need to be better at doing it theoretically.
Well, and you see now why you start seeing so many other combinations, still using the old chemotherapy, to try to get that initial shock, and then continue. How many combination trials are now with chemo and checkpoint inhibitors?
Yeah, but that being said, it's interesting. I mean, this comes up often on panels. The old mantra, of course, was you'll never get an immune response in advanced stage patients. Well, that's not true. And lung cancer is darn cold, and it's never going to work there. So much for that. So we're doing a lot of learning on the job still, for sure. I want to go back. I know a couple of you mentioned explicitly having COVID programs, but in the context of we're just some of us more than others, kind of coming out of a year plus dark days, has the success, the collaboration between industry, governments, other organizations, et cetera, in bringing, vaccines forward really rapidly, has that helped the story as you try to tell it about cancer vaccines? And not just RNA-based, vaccination approaches, just broadly, have you seen an opening up of the minds?
I think there's beginning to be approach that I think Andrew mentioned this, again, coming back to this idea of one of the breakthroughs is going to be better designs of clinical trials and better endpoints. And the speed with which the COVID vaccines went through the system showed that the system is actually more flexible than perhaps we would have believed at the beginning. And that when they put their mind to it, they can actually find regulatory pathways that make both sense for the safety side, but also for the efficacy side. And hopefully they will learn the same for the cancer vaccine.
Great, thanks. Andrew, you were...
Yeah, I think there's been a step up in the sophistication of the general audience. There's still plenty of room to go, I think. But what we've seen, obviously, is the protective power of neutralizing antibodies against SARS-CoV-2. And that's focused people's minds on vaccines and lipid nanoparticles and RNA, and that's all good. There's also some awareness that CD8s and neutralizing antibodies are distinct arms of the immune response, and they play different roles. I think some of the sophistication breaks down now because we don't have standard T cell assays. Many people don't differentiate between CD4s and CD8s. They just talk about T cells. So there's room for improvement on this dimension. But I think the general level of sophistication has increased. Of course, neutralizing antibodies are immaterial, as far as we know, to cancer immunotherapy. And therefore, what we've learned about the power of RNA does not translate into cancer immunotherapy. It's a fundamentally different value proposition, where our data suggests that CD8s remain the final common pathway of tumor destruction. And I agree with Carlos, obviously there is a role for CD4s in the generation of a potent CD8 response, as well as the generation of a potent antibody response. I mean, CD4s are referred to as the conductor of the immune orchestra for a reason. So it's not that I think any of us believe CD4s are unimportant, but most tumors do not express class 2 HLA. Therefore, you need destruction to be mediated. If it's direct destruction, it has to be mediated through class 1 recognition, which means CD8s. So the focus on CD8s obviously is interesting. And that's obviously the area where we've engaged with COVID-19, which is that, we have platforms, as you say, heterologous prime boost. We use an adenovirus. We use self-amplifying RNA. We can make really nice antibody responses, but we can also make potent CD8s. So why does that matter? Where does that matter? How does that help? These are kind of the interesting questions that we're wrestling with in COVID-19.
We're just running up against time. Any final comments from anyone?
Quickly, Jeff, I just want to say one thing on the COVID-19 story. I think it does help a little. Like we are seeing renewed interest in the space, generally speaking. And what's fascinating to me, if you look at the history of mRNA, from the 90s to 2 years ago, there was not a lot of success happening, and they figured out something in the formulation. Really, that's what's making the difference. Those new lipids in the formulation. And suddenly you have that overnight success. So I'm just, very hopeful for the cancer vaccine space that the same kind of thing is going to happen at one point. And then we're going to flip, really the sentiment over cancer vaccines. And suddenly a lot of people will find a lot of good reasons to believe, which is not the case today.
Well, I certainly hope so. Anyone else have any additional comments?
Yeah, maybe 1 minute. So I think we learned also from the COVID vaccine development that antibodies that target only one target, the virus can evolve and mutate very quickly. And we anticipated that with, I'm sure Andrew is the same, to generate or develop a multi-target approach against the virus and the variants. And that's the same for the tumors, I think. If we target only one antigens, we know that type of drugs will fail. So I think the highlights that there is now on T cells against the COVID-19 and the variants to cover the heterogeneity and the escape of the virus will be very easily translatable to the cancer. We all know that, but I think the field now will be able to appreciate that point too. That's very interesting.
Yeah. I think that's a great way to end, the idea that, hopefully vaccines and other pieces of the IO toolkit and quote unquote non-IO agents can really kind of help here to bring the plasticity, diversity, heterogeneity that is the immune system as a perfectly matched sparring partner with that very plastic and heterogeneous tumor. So, all right, I want to thank all of you, Carlos, Mike, Fred, Nicolas, Andrew, for a great discussion. Thank you very much and stay safe.
Thank you.
Thank you.
Thank you.
Thank
Thanks,
you.
everyone.
Bye.
Take care.
Bye.
Thanks.